Hello everyone, my name is Saroj Bhangade. Today date is the 9th February 2022. Today video topic is the origin of pharmacovigilance. Pharmacovigilance is a design in two word. Pharmacon is a Greek word to drug and medicinal substance, and second is a vigilance is a Latin word to the watch to keep watching. Pharmacovigilance is a drug safety and medicinal product. The definition of pharmacovigilance is the science and activities relating to the detection, assessment, understanding, prevention of the adverse effect or any other drug related problem. The history of pharmacovigilance during the 20th century, there were some serious adverse event associated with the medicinal product and drug. The first is anesthesia, chloroform anesthesia. The history of pharmacovigilance started in 169 years ago. On January 29, 1848, the young girl Huna Greener from the north of England dies after the receiving the chloroform anesthesia. Before the removal of infected toenail. Second, the sulfonamide disaster in 1937. There were 107 deaths in USA because of the sulfonamide elixir containing the diethylene glycol as a solvent. This solvent was the culprit was the dead. Consequently, the Federal Food, Drug and Cosmetic Act is established in 1938. Third is the thalidomide disaster in Germany 1961. The use of the thalidomide 46 country by women who is the pregnant. The symptom of the morning symptom is the nausea. Using the subsequently become a pregnant woman resulted in the biggest man-made disaster ever resulting is more than 10,000 children are born with these formalities and such as the phocomelia as well as the thousand of miscarriages and 1000 children are dies. The Frankins Casely is a food and drug administration reviewer. She is recently joined the FDA. She refused to authorize the evidence of the evidence regarding to the drug safety. Casely was the second woman to receive the president award for the distinct federal civilian service in 1962. Development of the pharmacovigilance. Thalidomide tragedy lead in Europe and other countries to other countries. The establish of the drug regulatory authorities. FDA started the systematic collection of the report on all type of the ADR. Government organization were instructed to set up the post marketing surveillance system as to pick any ADR early as possible, prevent or similar tragedy in future. The first step of the pharmacovigilance is the detection. Find out the ADR and clinical trial investigational detect the ADR in post marketing surveillance. Patient, physician, pharmacists are also reported. Second is the assessment. In this step, check out the ADR occur because due to the drug or any other reason of the ADR. Assessment make us from the ACP's healthcare professional. Third is a understanding. In this step, confirm the ADR occurred by the drug. Then the doctor and research scientists find out what are the ADR ADR like drug reaction or drug food reaction. Fourth is a prevention. To prevent and stop the ADR, proactively report the regulated authorities and take a precaution would help in the prevent or adverse effect in future. The aim and objective of the pharmacovigilance is the first is the public First is the patient care, to improve the patient care and safety in relation to the medicine and all medical and paramedical invention. Second is the public health, to improve the public health and safety of the relation use of the medicine. Third is the risk and benefit assessment, to contribute the assessment of the harmful effectiveness and risks of medicine. Fourth is the communication, to promote the understanding, clinical training and effective communication to the health professional and public. The second is the regulatory bodies. Regulatory bodies is the government concern which is mainly work on the rules and guideline and the regulation, observe the compliance. Regulatory bodies which mainly help to the followed by the regulation guideline, rules, regulation and guideline. Regulatory bodies different countries is the US is the FDA, Food and Drug Administration. Second is the India, CDSCO, Central Drug Standard Control Organization. Third is the UK, MHRA, Medicine of Healthcare Product Medicine of Healthcare Product Regulatory Agencies. Australia is the TGA, Therapeutic Good Administration. Japan is the MHLW, Ministry of Health, Labour and Welfare. Europe is the EMA, European Medicine Agency. The area reporting form. The reporting form in India is the suspected adverse drug reaction reporting form. The reporting form in UK is the yellow card system. Third is the reporting form is the US is the MedWatch. The MedWatch are the two subtype. First is the MedWatch 3500, used for the general public, and second is the MedWatch 3500A, used for the healthcare professional. Adverse report system is the adverse event reporting form. FDA as well reporting system, fully computerized and fully computerized automatic data and spontaneous report, and fully spontaneous and spontaneous report. Second is the drug discovery and clinical trial. The drug discovery mean by the pharmaceutical, what is the drug? Mean by the pharmaceutical agent or medicinal product that used for the treatment or prevention of disease. Drug discovery is begin in the laboratories, identify the cellular genetic factor that play the scientific role of the disease. Drug discovery involved in the two step. First is the drug discovery and second is the drug development. Drug discovery is the take approximately 10 to 15 years and lots of money for the new drug. In ancient time, drug have been discovered either by the identified active ingredient in traditional remedies by the discovery, but now this day drug would be say now by this day discovered by the disease of the basis of the disease. Aim of the drug discovery is the drug would be safe, effective and less toxic effect. Drug discovery and drug development involving six steps. First is the target identification, second is the target validation, third is the lead optimization, and fourth is the lead compound, fifth is the preclinical, and sixth is the clinical trial. First is the target identification. Firstly, we have identified the disease or problem for which find out the disease, and in this step identify the disease organ involved in the that particular organ involved in the defective due to the disease. It can be any organ, tissues, protein, and some receptor involved in the in this step, nuclear receptor, ion channel and enzyme. Second is the target validation. Now in this step choose the target and analyze and validate the information is correct or not, and also check the drug ability to drug ability can target the actually bind to the drug. Third is the lead identification. In this step identification of the drug that actually used to the cure and cure that this is the lead compound can be synthesized in laboratories, identify the drug which is synthesized, which is have potential bind to the identifies. Fourth is the lead optimization. In this it is a final drug candidate we get after the drug identifies phase. In this, drug candidate passes through the screening process and uh, only five compound are been selected in drug optimization step. In this opted five compound are called as a lead compound, and then further the clinical trial, the development phase. In this phase, drug is developed in for the market use. It is involved in two steps. First is the preclinical and second is the clinical. First is the preclinical. Before the testing in human on before the testing a drug on people, we have to confirm the drug is safe and does not give the harmful effect of the for the human. For this purpose, firstly we tested in animal. The aim of the preclinical to check the safety and effective of the drug, to check the drug pharmacokinetic property, to check the toxicity and toxicology, and also check the evolution therapeutic index. The two type of the preclinical trial. First is the in vitro and second is the in vivo. In vitro is the experiment conducted in the outside the animal. We collect the animal tissue, plasma, etc. And in vivo is the experiment conducted inside the animal. Animal use the introducing the drug inside the animal. The animal use of the preclinical is a mice, rat, monkey, dogs. After all this, we collect the information. Now this study provide the details in information is sent to INDA, investigation new drug application. In this after we preclinical study success and the NDA approve the drug. The it further forwarded to the clinical trial. Both drug discovery and preclinical phases are the approximately 6 years. Second is the clinical phases. In this step test is done in a human to involve in the 5 phases. Phase 0, phase first, second, phase third, phase 4. Phase 0 is the microdosing. Study the new microdose to the derive the pharmacokinetic information in human before the under the testing first phase. The study is called the phase 0. Microdose is the microdose is the less than 1 by 100 of the dose. Advantage of the phase 0 is the less chance to adverse effect and short duration, less number of volunteers, reduce the cost of the development of the drug and time. Limitation of the phase 0 is the mainly study the based on the pharmacokinetic parameter. The laboratory parameter are very limited and expensive, depend on the bioavailability and bioequivalent lab. Phase first is the testing in human subject 20 to 80 healthy volunteer and patient volunteer. Patient volunteer like AIDS therapy and cancer patient. The main purpose of this phase is to check the safety and side effect of the drug. The limitation of the phase first is the perform the extrapolated heterogeneous marketplace. Phase second is the therapeutic exploratory trial of the consist of the 20 to 300 subject to confirm the effective, monitor side effect and safety. Length of this phase is 6 months to several years. Phase second are the two subtypes. First is the phase second A is a design access to dosing requirement, and phase second B is a design the study efficacy. Phase third is it is a therapeutic confirmatory trial, target the population and service to 1000 to 3000 patients. Duration of this phase is up to 5 years, and phase 3 is phase 3 are two subtype. Phase third A is to get the sufficient and significant data, and phase second B is after the NDA by approval and launch after the completing first phase third trial drug in undergoes for the approval to FDA. After FDA approval, the product is launched into the market. Phase 4 this phase is the post marketing surveillance, no fixed duration, and patient population perform the quality of the live trial and collect all long term safety inform. ultimate goal of Ultimate goal of the drug discovery is the drug is approved. That's about all drug discovery and drug development and pharmacovigilance, origin of pharmacovigilance and regulatory bodies. Thank you.